Right, this is a quick walkthrough of mapping sequence variants at the genomic level onto protein structures with Jalview. So I've cleared all the windows in Jalview just by using Windows close all there. So I've got a clean desktop to start with. So I'll go on to File. I'm going to fetch a sequence, and I'm going to fetch that sequence from Ensemble. And click OK there to go to the database, and I'm going to put in a UniProt identifier for the protein sequence I'm interested in. That's 30419. This is N-myristoyl transferase. Click OK, and this will go and fetch the sequence entry from Ensemble. Takes just a few minutes, pulls back the data, and should give me a nice sequence. Just a moment or two, and it should be there. There we go. Okay, so what it's done here, it's pulled back the gene entry. I see lots of entries there, and all the transcript information, all the different transcripts. So if I go to View, I can pull up the overview window, and that will show me, and that bit bigger, this shows me all the transcripts mapped onto the sequence. Now what it has done here, let's just make this window a bit bigger, close the overview window. This little blue bit there tells me that what it's done is it's hidden from the genomic sequence, the ENSG sequence, it's hidden all the introns. It's just showing me the exonic sequences, the ones that appear in the transcripts. So this makes it actually a viable sequence to look at. And as we go through, we can see the transcripts, we can see the variants mapped onto them, and there's a variant transcript there, and all the exons are present as different colors. This we'll see on the protein structure in just a moment. Now what we're going to do is to map this through to the protein, go into Calculate, get cross-references, and it's going to cross-reference through to UniProt here. So we'll click UniProt. Up comes a two-pane window. At the bottom here we've got the protein sequence, and we can see we're in exon from exon 1, and as we move through that you can see the codon change up here, and you see parallel in the protein sequence, maps of conservation and so on. Now this is quite a small window. I'm just going to stretch this one a wee bit, because I want to take this sequence here. It's got PDB sequences. You just see those at the bottom there, PDB structures. I can right click, go to 3D structure data. This now pulls me back a list of all the crystal structures that are related to this. The first one is going to be as good as anything. We'll go with that. And there we go, crystal structure of human type 1 N-myristoyl transferase. Let's go with that and view that. So this comes back and it displays it using the JMOL applet. It is possible to get this into Chimera as well, directly from Jalview, but in this case we're just going to put it into JMOL, and I'm just going to make this a little bigger, push it up into the corner there, and we can just grab on that left mouse button, drag that, and then we can spin it around quite happily. You can see the colors actually map through. So where it's blue, green, and so on, that matches back here where we've got the exons for that particular sequence. We can see the variants. Let's go into the light blue ones here and go back to our window with the JMOL view in. And you see, as we move over, it might be a bit tricky to see, but as we move over the sequence, the amino acids do pop up on Jalview instead of just the ribbon. You can see them popping up through there. Let's just spin that round a bit, make it potentially a bit clearer. See if we can get a nice view there. Yeah, there you go. You can see them popping up as we go. So we can go directly from that variant, and we can see what that variant is, C to T variant, through. You can see what the exon is. It maps on exactly, so you can see that's going to be an ACG to ATG, so threonine to methionine variant, and you can see exactly where that's popping up on the protein structure here. There we go, just there, that threonine there. So we've gone from sequence variants straight through to structure variants from genome to protein structure with almost no effort at all.